So, hi everyone, and thank you for joining me. My name is Claire Schraidt, and I just finished up my master's degree here at Purdue. And the lab I work in works both in the Department of Forestry and Natural Resources and the Department of Ecology and Evolutionary Biology. And we use genetics to think about how fish in the Great Lakes should be managed. And I think this is pretty cool because in the summer, I get to spend my time out on boats on Lake Michigan, as you can see in this picture, catching some really cool fish. And in the winter, I get to do some fun laboratory work and a little bit of bioinformatics. So let's get started. So who can spot the differences between these fish? Are there any differences? Maybe size, color, number of fish? So this is actually a trick question. On the surface, there really isn't any difference between any of these fish. And this is actually a real world challenge that fisheries managers face when they're trying to identify populations. Particularly for fish species, we can't observe them in their environment, so we really don't know who's related to one another or how they're interacting with each other. And so what if I told you that there are three distinct populations here, and you needed to manage each one in a different way? How would you decide to group them just based on this photo? So let's put down some lines. Maybe we can try organizing these units spatially. So let's make the top right maybe one management unit, and maybe let's just split that bottom chunk into two. Okay, so now that we have our management units drawn, let's see how we did. So we didn't do a really good job at this, but how are we supposed to do better when all the populations look like this? So luckily, using genetics, we can figure out how fish are related to each other based off their DNA. And you guys can sort of think of this as 23andMe, but for fish. And using genetics, we can start to parse apart what those populations actually look like, despite the fact that fish might look the same on the surface, just with the naked eye. And I use this method to figure out how populations of yellow perch are structured in Lake Michigan. So in this top left corner, you see a yellow perch. And they're a popular fish species for recreational fishermen, and they're still commercially fished in some parts of the Great Lakes. However, this population crashed in the late 80s, and despite our best management efforts, as you can see here, even through 2015 through now, these populations are still not doing very well. And in order to make sure we're doing the best possible job of managing yellow perch, we want to delineate populations in Lake Michigan and make sure we really know what we're trying to manage. And we accomplished this using a couple of steps. So step one, we collected Lake Michigan individuals from all around Lake Michigan. Step two, we used genetic information from these individuals to figure out how they're related to one another. And step three, profit. Just kidding. What we found is that yellow perch and green bay are genetically distinct from the rest of Lake Michigan, and they'll probably need a specific management plan. Future work on this project will include trying to further differentiate population differences among fish in the main basin, but that's going to be someone else's research project. Other people in my lab look at different species in the Great Lakes, including Atlantic salmon, pink salmon, and sea lamprey. And we really have a good time out in the field collecting specimens, extracting DNA in the lab, and of course, using our research to inform real-world management decisions. So thanks guys so much for listening to my quick little talk, and I really encourage you to feel free to drop me a question and let me know what you think. Thanks.